Cytokinin is one of the major plant growth regulator. The first isolated cytokinin was named kinetin, and the first isolated naturally occurring cytokinin was zeatin. So, if you see any question where zeatin is used, so don't get confused. It is a type of cytokinin. They all are substituted adenine, and common active cytokine includes zeatin and isopentenyl adenine. The main function of cytokinin includes it inhibits leaf senescence, which is the falling of leaf with the age of a plant, and it promotes cell division, hence name cytokinin. That is the it promotes cytokinesis, which means cell division. Therefore, it is majorly present in meristematic regions or growing tissue where cytokinin is majorly occur. Then it promotes auxiliary bud development and inhibit apical dominance. This function of cytokinin is just opposite to that of auxin, which inhibit auxiliary bud development and induce apical dominance. Next, the biosynthesis and transport of cytokinin. Synthesis it is synthesized in roots, and the transport it is transported to shoot via xylem. Then how it synthesized? Zeatin is synthesized from isopentenyl pyrophosphate with the help of AMP. It get converted to isopentenyl AMP. Then removal of phosphate from isopentenyl AMP form isopentenyl adenosine. Then removal of ribose sugar, ribose group from isopentenyl adenosine form isopentenyl adenine, and finally the oxidation of isopentenyl adenine form zeatin. Now the most important part here is signaling. Its signaling is very similar to two component system of bacteria. In a standard two component system, it is there are two components are present, that is sensor histidine kinase and a response regulator. A response regulator has two domains, a receiver domain and a effector domain. So what actually occur is this sensor is present on the membrane, and when a ligand binds from the extracellular medium, it get activated, and this histidine kinase get autophosphorylated with the help of ATP. Then this phosphate is transferred to the receiver domains. Aspartate receiver domain has aspartate in it, so it get transferred to aspartate. Then from receiver domain it finally transferred to the effector domain, from where gene regulation occur. Then how it is similar to the Arabidopsis multi-step phosphorylase pathway. It is also an advanced version of two component system, and we use Arabidopsis in plants. It is a model of plant development, like we have Drosophila in animal development. So here we have three things. First one is the hybrid sensor kinase. Here we call it AHK. It is a receptor of cytokinin. So AHK is Arabidopsis histidine kinase. So in any protein we use Arabidopsis. So phosphotransfer protein, it is also known as AHP here. AHP is Arabidopsis histidine phosphotransfer protein. Otherwise, simply it is also known as HTP, which is histidine phosphotransfer protein. And lastly, we had the response regulator. So here it occurs in endoplasmic reticulum. It is present in the receptor is present in the form of dimer, and inside the endoplasmic reticulum membrane is a chase domain of this receptor. Chases cyclase histidine kinase associated sensor extracellular. Here cytokinin binds, and when cytokinin bind to the chase domain, this get activated, and the histidine get autophosphorylated with the help of ATP. Then the phosphate is transferred to the receiver domain, which has aspartate, and finally the phosphate from here is transferred to AHP, which is Arabidopsis histidine phosphotransfer protein. It functions as a signaling shuttle between cytoplasm and nucleus, as it transfers the phosphate from cytoplasm to nucleus response regulator. The response regulator is called here Arabidopsis response regulator, and finally it shows the gene regulation. So lastly, we have two classes of ARRs. Arabidopsis response regulators, a type A and type B. Type B is the main transcriptional activator. It has a receiver domain and output domain. Output domain has DNA binding and transcription activating domain also. So when output domain is activated in type B, it phosphorylated type A and type A also get activated, and it show its function. So type A is a negative regulator of cytokinin. It has only a single receiver domain. So they both functions in opposite. They are antagonists to each other. Lastly, the physiological effects of cytokinin. So revising the physiological effect, it helps in cell division and morphogenesis. This comes in many questions, many exams, like how it does. So auxin to cytokinin ratio is, if it is more, so root formation takes place, and the vice versa occurs. That is, if cytokinin to auxin ratio is more, then shoot is formed. Then it inhibits apical dominance, and it does chloroplast movement. That is it from chloroplast from etioplast. And lastly, it delay senescence. Thank you.